Chapter 15 Intracellular Compartments and Transport. At any one time, a typical eukaryotic cell carries out thousands of different chemical reactions, many of which are mutually incompatible. One series of reactions makes glucose, for example, while another breaks down glucose. Some enzymes synthesize peptide bonds, whereas others hydrolyze them, and so on. Indeed, if the cells of an organ such as the liver are broken apart and their contents mixed together in a test tube, chemical chaos results, and the cell's enzymes and other proteins are quickly degraded by their own proteolytic enzymes. For a cell to operate effectively, the different intracellular processes that occur simultaneously must somehow be segregated. Cells have evolved several strategies for isolating and organizing their chemical reactions. One strategy used by both prokaryotic and eukaryotic cells is to aggregate the different enzymes required to catalyze a particular sequence of reactions into large, multi-component complexes. Such complexes are used, for example, in the synthesis of DNA, RNA, and proteins. A second strategy, which is most highly developed in eukaryotic cells, is to confine different metabolic processes and the proteins required to perform them within different membrane-enclosed compartments. As discussed in Chapter 11 and 12, cell membranes provide selectively permeable membranes through which the transport of most molecules can be controlled. In this chapter, we consider the strategy of compartmentalization and some of its consequences. In the first section, we describe the principal membrane-enclosed compartments, or membrane-enclosed organelles of eukaryotic cells, and briefly consider their main functions. In the second section, we discuss how the protein composition of the different compartments is set up and maintained. Each compartment contains a unique set of proteins that have to be transferred selectively from the cytosol where they are made to the compartment in which they are used. This transfer process, called protein sorting, depends on signals built into the amino acid sequence of the proteins. In the third section, we describe how certain membrane-enclosed compartments in a eukaryotic cell communicate with one another by forming small membrane-enclosed sacs, or vesicles. These pinch off from one compartment, move through the cytosol, and fuse with another compartment in a process called Vesicular Transport. In the last two sections, we discuss how this constant vesicular traffic also provides the main routes for releasing proteins from the cell by the process of exocytosis, and for importing them by the process of endocytosis. Membrane-enclosed organelles. Whereas a prokaryotic cell usually consists of a single compartment, the cytosol, enclosed by the plasma membrane, eukaryotic cells are elaborately subdivided by internal membranes. These membranes create enclosed compartments in which sets of enzymes can operate without interference from reactions occurring in other compartments. When a cross-section through a plant or an animal cell is examined in the electron microscope, numerous small membrane-enclosed sacs, tubes, spheres, and irregularly shaped structures can be seen, often arranged without much apparent order. Figure 15.1. These structures are all distinct membrane-enclosed organelles, or parts of such organelles, each of which contains a unique set of large and small molecules and carries out a specialized function. In this section, we review these functions and discuss how different membrane-enclosed organelles may have evolved. Eukaryotic cells contain a basic set of membrane-enclosed organelles. The major membrane-enclosed organelles of an animal cell are illustrated in Figure 15.2, and their functions are summarized in Table 15.1. These organelles are surrounded by the cytosol, which is enclosed by the plasma membrane. The nucleus is generally the most prominent organelle in eukaryotic cells. It is surrounded by a double membrane, known as a nuclear envelope, and communicates with the cytosol via nuclear pores that perforate the envelope. The outer nuclear membrane is continuous with the membrane of the endoplasmic reticulum, ER, a system of interconnected sacs and tubes of membrane that often extends throughout most of the cell. The ER is the major site of synthesis of new membranes in the cell. Large areas of the ER have ribosomes attached to the cytosolic surface and are designated rough endoplasmic reticulum, rough ER. The ribosomes are actively synthesizing proteins that are delivered into the ER lumen or ER membrane. The smooth endoplasmic reticulum, smooth ER, lacks ribosomes. It is scanty in most cells but is highly developed for performing particular functions in others. For example, it is the site of steroid hormone synthesis in cells of the adrenal gland and the site where a variety of organic molecules, including alcohol, are detoxified in liver cells. In many eukaryotic cells, the smooth ER also sequesters Ca2+ from the cytosol. The release and reuptake of Ca2+ from the ER is involved in the rapid response to many extracellular signals, as discussed in chapters 12 and 16. The Golgi apparatus, which is usually situated near the nucleus, receives proteins and lipids from the ER, modifies them, and then dispatches them to other destinations in the cell. Small sacs of digestive enzymes called lysosomes degrade worn-out organelles, as well as macromolecules and particles taken into the cell by endocytosis. On their way to lysosomes, endocytosed materials must first pass through a series of compartments called endosomes, which sort the ingested molecules and recycle some of them back to the plasma membrane. Peroxisomes are small organelles enclosed by a single membrane. They contain enzymes used in a variety of oxidative reactions that break down lipids and destroy toxic molecules. Mitochondria and, in plant cells, chloroplasts are each surrounded by a double membrane and are the sites of oxidative phosphorylation and photosynthesis, respectively, discussed in chapter 14. Both contain membranes that are highly specialized for the production of ATP. Many of the membrane-enclosed organelles, including the ER, Golgi apparatus, mitochondria, and chloroplasts, are held in their relative locations in the cell by attachment to the cytoskeleton, especially to microtubules. Cytoskeletal filaments provide tracks for moving the organelles around and for directing the traffic of vesicles between them. These movements are driven by motor proteins that use the energy of ATP hydrolysis to propel the organelles and vesicles along the filaments, as discussed in chapter 17. On average, the membrane-enclosed organelles together occupy nearly half the volume of a eukaryotic cell, Table 15.2, and the total amount of membrane associated with them is enormous. In a typical mammalian cell, for example, the area of the endoplasmic reticulum membrane is 20 to 30 times greater than that of the plasma membrane. In terms of its area and mass, the plasma membrane is only a minor membrane in most eukaryotic cells. Much can be learned about the composition and function of an organelle once it has been isolated from other cell structures. For the most part, organelles are far too small to be isolated by hand, but it is possible to separate one type of organelle from another by differential centrifugation, described in panel 4.4, pages 164 to 165. Once a purified sample of one type of organelle has been obtained, the organelle's proteins can be identified. In many cases, the organelle itself can be incubated in a test tube under conditions that allow its functions to be studied. Isolated mitochondria, for example, can produce ATP from the oxidation of pyruvate to CO2 and water, provided they are adequately supplied with ADP and O2. Membrane-enclosed organelles evolved in different ways. In trying to understand the relationships between the different compartments of a modern eukaryotic cell, it is helpful to consider how they might have evolved. The compartments probably evolved in stages. The precursors of the first eukaryotic cells are thought to have been simple microorganisms, resembling bacteria, which had a plasma membrane but no internal membranes. The plasma membrane in such cells would have provided all membrane-dependent functions, including ATP synthesis and lipid synthesis, as does the plasma membrane in most modern bacteria. Bacteria can get by with this arrangement because of their small size and thus their high surface-to-volume ratio. Their plasma membrane area is sufficient to sustain all the vital functions for which membranes are required. Present-day eukaryotic cells, however, have volumes 1,000 to 10,000 times greater than that of a typical bacterium, such as E. coli. Such a large cell has a small surface-to-volume ratio and presumably could not survive with a plasma membrane as its only membrane. Thus, the increase in size typical of eukaryotic cells probably could not have occurred without the development of internal membranes. Membrane-enclosed organelles are thought to have arisen in evolution in at least two ways. The nuclear membranes and the membranes of the ER, Golgi Apparatus, endosomes, and lysosomes are believed to have originated by invagination of the plasma membrane. Figure 15.3. These membranes and the organelles they enclose are all part of what is collectively called the endomembrane system. As we discuss later, the interiors of these organelles, with the exception of the nucleus, communicate extensively with one another and with the outside of the cell by means of small vesicles that bud off from one of these organelles and fuse with another. Consistent with this proposed evolutionary origin, the interiors of these organelles are treated by the cell in many ways as extracellular, as we shall see. The hypothetical scheme shown in Figure 15.3 would also explain why the nucleus is surrounded by two membranes. Although membrane invagination is rare in present-day bacteria, it does occur in some photosynthetic bacteria in which the regions of the plasma membrane containing the photosynthetic apparatus are internalized, forming intracellular vesicles. Mitochondria and chloroplasts are thought to have originated in a different way. They differ from all other organelles in that they possess their own small genomes and can make some of their own proteins, as discussed in Chapter 14. The similarity of these genomes to those of bacteria and the close resemblance of some of their proteins to bacterial proteins strongly suggest that mitochondria and chloroplasts evolved from bacteria that were engulfed by primitive eukaryotic cells with which they initially lived in symbiosis. Figure 15.4. As might be expected from their origins, mitochondria and chloroplasts remain isolated from the extensive vesicular traffic that connects the interiors of most of the other membrane-enclosed organelles to one another and to the outside of the cell. Having briefly reviewed the main membrane-enclosed organelles of the eukaryotic cell, we turn now to the question of how each organelle acquires its unique set of proteins. Protein sorting. Before a eukaryotic cell reproduces by dividing in two, it has to duplicate its membrane-enclosed organelles. A cell cannot make these organelles from scratch. It requires information and materials contained in the organelle itself. Thus, most of the organelles are formed from pre-existing organelles, which grow and then divide. As cells grow, membrane-enclosed organelles enlarge by incorporation of new molecules. The organelles then divide and during cell division are distributed between the two daughter cells. Organelle growth requires a supply of new lipids to make more membrane and a supply of the appropriate proteins, both membrane proteins and the soluble proteins that will occupy the interior of the organelle. Even in cells that are not dividing, proteins are being produced continually. These newly synthesized proteins must be accurately delivered to their appropriate organelle, some for eventual secretion from the cell, and some to replace organelle proteins that have been degraded. Directing newly made proteins to their correct organelle is therefore necessary for a cell to be able to grow, divide, and function properly. For some organelles, including the mitochondria, chloroplasts, and the interior of the nucleus, proteins are delivered directly from the cytosol. For others, including the Golgi Apparatus, lysosomes, endosomes, and the nuclear membranes, proteins and lipids are delivered indirectly via the ER, which is itself a major site of lipid and protein synthesis. Proteins enter the ER directly from the cytosol. Some are retained there, but most are transported by vesicles to the Golgi apparatus and then onward to other organelles or the plasma membrane. In this section, we discuss the mechanisms by which proteins directly enter membrane-enclosed organelles from the cytosol. Proteins made in the cytosol are dispatched to different locations in the cell according to specific address labels that they contain in their amino acid sequence. Once at the correct address, the protein enters the organelle. Proteins are imported into organelles by three mechanisms. The synthesis of virtually all proteins in the cell begins on ribosomes in the cytosol. The exceptions are the few mitochondrial and chloroplast proteins that are synthesized on ribosomes inside these organelles. Most mitochondrial and chloroplast proteins, however, are made in the cytosol and subsequently imported. The fate of any protein molecule synthesized in the cytosol depends on its amino acid sequence, which can contain a sorting signal that directs the protein to the organelle in which it is required. Proteins that lack such signals remain as permanent residents in the cytosol. Those that possess a sorting signal move from the cytosol to the appropriate organelle. Different sorting signals direct proteins into the nucleus, mitochondria, chloroplasts in plants, peroxisomes, and the ER. When a membrane-enclosed organelle imports proteins from the cytosol or from another organelle, it faces a problem. How can it draw the protein across membranes that are normally impermeable to hydrophilic macromolecules? This task is accomplished in different ways for different organelles. 1. Proteins moving from the cytosol into the nucleus are transported through the nuclear pores that penetrate the inner and outer nuclear membranes. The pores function as selective gates that actively transport specific macromolecules, but also allow free diffusion of smaller molecules. Mechanism 1 in Figure 15.5. 2. Proteins moving from the cytosol into the ER, mitochondria, or chloroplasts are transported across the organelle membrane by protein translocators located in the membrane. Unlike transport through nuclear pores, the transported protein molecule must usually unfold in order to snake through the membrane. Mechanism 2 in Figure 15.5. Bacteria have similar protein translocators in their plasma membrane, which they use to export proteins from their cytosol. 3. Proteins moving from the ER onward and from one compartment of the endomembrane system to another are transported by a mechanism that is fundamentally different from the other two. These proteins are ferried by transport vesicles, which become loaded with a cargo of proteins from the interior space, or lumen, of one compartment as they pinch off from its membrane. The vesicles subsequently discharge their cargo into a second compartment by fusing with its membrane. Mechanism 3 in Figure 15.5. In the process, membrane lipids and membrane proteins are also delivered from the first compartment to the second. Signal sequences direct proteins to the correct compartment. The typical sorting signal on proteins is a continuous stretch of amino acid sequence, typically 15 to 60 amino acids long. This signal sequence is often, but not always, removed from the finished protein once it has been sorted. Some of the signal sequences used to specify different destinations in the cell are shown in Table 15.3. Signal sequences are both necessary and sufficient to direct a protein to a particular organelle. This has been shown by experiments in which the sequence is either deleted or transferred from one protein to another by genetic engineering techniques, discussed in Chapter 10. Deleting a signal sequence from an ER protein, for example, converts it into a cytosolic protein, while placing an ER signal sequence at the beginning of a cytosolic protein redirects the protein to the ER. Figure 15.6. The signal sequences specifying the same destination can vary greatly even though they have the same function. Physical properties such as hydrophobicity or the placement of charged amino acids often appear to be more important for the function of these signals than the exact amino acid sequence. Proteins enter the nucleus through nuclear pores. The nuclear envelope encloses the nuclear DNA and defines the nuclear compartment. It is formed from two concentric membranes. The inner nuclear membrane contains proteins that act as binding sites for the chromosomes, discussed in Chapter 5, and provide anchorage for the nuclear lamina, a finely woven meshwork of protein filaments that lines the inner face of this membrane and provides a structural support for the nuclear envelope, discussed in Chapter 17. The composition of the outer membrane closely resembles the membrane of the ER, with which it is continuous. Figure 15.7. The nuclear envelope in all eukaryotic cells is perforated by nuclear pores that form the gates through which all molecules enter or leave the nucleus. Traffic occurs in both directions through the pores. Newly made proteins destined for the nucleus enter from the cytosol. Movie 15.1. RNA molecules, which are synthesized in the nucleus, and ribosomal subunits, which are assembled in the nucleus, are exported. Messenger RNA molecules that are incompletely spliced are not exported from the nucleus, suggesting that nuclear transport serves as a final quality control step in mRNA synthesis and processing, discussed in Chapter 7. A nuclear pore is a large, elaborate structure composed of about 30 different proteins. Figure 15.8. Each pore contains water-filled passages through which small water-soluble molecules can pass freely and non-selectively between the nucleus and the cytosol. Many of the proteins that line the nuclear pore contain extensive, unstructured regions, which are thought to form a disordered tangle, much like a kelp bed in the ocean. This jumbled meshwork fills the center of the channel, preventing the passage of large molecules, but allowing smaller molecules to slip through. Larger molecules, such as RNAs and proteins and macromolecular complexes, must display an appropriate sorting signal to pass through the nuclear pore. The signal sequence that directs a protein from the cytosol into the nucleus, called a nuclear localization signal, typically consists of one or two short sequences containing several positively charged lysines or arginines. See Table 15.3. Cytosolic proteins called nuclear transport receptors bind to the nuclear localization signal on newly synthesized proteins destined for the nucleus. These receptors help direct the new protein to a nuclear pore by interacting with the tentacle-like fibrils that extend from the rim of the pore, figure 15.9. During transport, the nuclear transport receptors grab onto repeated amino acid sequences within the tangle of nuclear pore proteins, pulling themselves from one to the next to carry their cargo protein into the nucleus. Once the protein has been delivered, the nuclear transport receptor is returned to the cytosol via the nuclear pore for reuse. See figure 15.9. Like any process that creates order, importing proteins into the nucleus requires energy. In this case, the energy is provided by GTP hydrolysis, which drives nuclear transport in the appropriate direction. Figure 15.10. Nuclear pore proteins operate this molecular gate at an amazing speed, pumping macromolecules in both directions through each pore. Nuclear pores transport proteins in their fully folded conformation and transfer ribosomal components as assembled particles. This feature distinguishes the nuclear transport mechanism from the mechanisms that transport proteins into other organelles. Proteins have to unfold during their transport across the membranes of other organelles such as mitochondria, chloroplasts, and the ER, as we discuss next. Proteins unfold to enter mitochondria and chloroplasts. Both mitochondria and chloroplasts are surrounded by inner and outer membranes, and both organelles specialize in the synthesis of ATP. Chloroplasts also contain a third membrane system, the thylakoid membrane, discussed in Chapter 14. Although both organelles contain their own genomes and make some of their own proteins, most mitochondrial and chloroplast proteins are encoded by genes in the nucleus and are imported from the cytosol. These proteins usually have a signal sequence at their N-terminus that allows them to enter their specific organelle. Proteins destined for either organelle are translocated simultaneously across both the inner and outer membranes at specialized sites where the two membranes are in contact with each other. Each protein is unfolded as it is transported, and its signal sequence is removed after translocation is complete. Figure 15.11. Chaperone proteins, discussed in Chapter 4, inside the organelles, help to pull the protein across the two membranes and to refold the protein once it is inside. Subsequent transport to a particular site within the organelle, such as the inner or outer membrane or the thylakoid membrane and chloroplasts, usually require further sorting signals in the protein, which are often only exposed after the first signal sequence has been removed. The insertion of transmembrane proteins into the inner membrane, for example, is guided by signal sequences in the protein that start and stop the transfer process across the membrane, as we describe later for the insertion of transmembrane proteins in the ER membrane. The growth and maintenance of mitochondria and chloroplasts require not only the import of new proteins, but also the incorporation of new lipids into their membranes. Most of their membrane phospholipids are thought to be imported from the ER, which is the main site of lipid synthesis in the cell. Phospholipids are transported individually to these organelles by water-soluble lipid-carrying proteins that extract a phospholipid molecule from one membrane and deliver it to another. Thanks to these proteins, the different cellular membranes are able to retain their characteristic lipid composition. Proteins enter the endoplasmic reticulum while being synthesized. The endoplasmic reticulum, ER, is the most extensive membrane system in a eukaryotic cell. Figure 15.12a. Unlike the organelles discussed so far, it serves as an entry point for proteins destined for other organelles, as well as for the ER itself. Proteins destined for the Golgi apparatus, endosomes, and lysosomes, as well as proteins destined for the cell surface, all first enter the ER from the cytosol. Once inside the ER or embedded in the ER membrane, individual proteins will not re-enter the cytosol during their onward journey. They will be ferried by transport vesicles from organelle to organelle, and in some cases from organelle to the plasma membrane or to the exterior of the cell. Two kinds of proteins are transferred from the cytosol to the ER. 1. Water-soluble proteins are completely translocated across the ER membrane and are released into the ER lumen. 2. Prospective transmembrane proteins are only partly translocated across the ER membrane and become embedded in it. The water-soluble proteins are destined either for secretion by release at the cell surface or for the lumen of an organelle. The transmembrane proteins are destined to reside in either the ER membrane, the membrane of another organelle, or the plasma membrane. All of these proteins are initially directed to the ER by an ER signal sequence, a segment of 8 or more hydrophobic amino acids. See Table 15.3, page 502. That is also involved in the process of translocation across the membrane. Unlike the proteins that enter the nucleus, mitochondria, chloroplasts, and peroxisomes, most of the proteins that enter the ER begin to be threaded across the ER membrane before the polypeptide chain has been completely synthesized. This requires that the ribosome synthesizing the protein be attached to the ER membrane. These membrane-bound ribosomes coat the surface of the ER, creating regions termed rough endoplasmic reticulum because of the characteristic beaded appearance when viewed in an electron microscope. Figure 1512b. There are, therefore, two separate populations of ribosomes in the cytosol. Membrane-bound ribosomes are attached to the cytosolic side of the ER membrane and outer nuclear membrane, and are making proteins that are being translocated into the ER. Free ribosomes are unattached to any membrane and are making all of the other proteins encoded by the nuclear DNA. Membrane-bound ribosomes and free ribosomes are structurally and functionally identical. They differ only in the proteins they are making at any given time. When a ribosome happens to be making a protein with an ER signal sequence, the signal sequence directs the ribosome to the ER membrane. As an mRNA molecule is translated, many ribosomes bind to it, forming a polyribosome, discussed in Chapter 7. In the case of an mRNA molecule encoding a protein with an ER signal sequence, the polyribosome becomes riveted to the ER membrane by the growing polypeptide chains, which have become inserted into the membrane. Figure 1513. Soluble proteins are released into the ER lumen. The ER signal sequence is guided to the ER membrane with the aid of at least two protein components. 1. A signal recognition particle, SRP, present in the cytosol, which binds to the ER signal sequence when it is exposed on the ribosome. And 2. An SRP receptor, embedded in the membrane of the ER, which recognizes the SRP. Binding of an SRP to a signal sequence causes protein synthesis by the ribosome to slow down until the ribosome and its bound SRP locate an SRP receptor on the ER. After binding to its receptor, the SRP is released and protein synthesis recommences, with the polypeptide now being threaded into the lumen of the ER through a translocation channel in the ER membrane. Figure 15.4. Thus, the SRP and SRP receptor function as molecular matchmakers, connecting ribosomes that are synthesizing proteins containing ER signal sequences to available ER translocation channels. In addition to directing proteins to the ER, the signal sequence, which for soluble proteins is almost always at the N-terminus, functions to open the translocation channel. The signal peptide remains bound to the channel while the rest of the protein chain is threaded through the membrane as a large loop. At some stage during translocation, the signal sequence is cleaved off by a signal peptidase located on the luminal side of the ER membrane. The signal peptide is then released from the translocation channel and rapidly degraded. Once the C-terminus of the protein has passed through the membrane, the protein is released into the ER lumen. Figure 1550. Start and stop signals determine the arrangement of a transmembrane protein in the lipid bilayer. Not all proteins that enter the ER are released into the ER lumen. Some remain embedded in the ER membrane as transmembrane proteins. The translocation process for such proteins is more complicated than it is for soluble proteins, as some parts of the polypeptide chain must be translocated clear across the lipid bilayer, while others remain fixed in the membrane. In the simplest case, that of transmembrane protein with a single membrane spanning segment, the N-terminal signal sequence initiates translocation, just as for a soluble protein. But the transfer process is halted by an additional sequence of hydrophobic amino acids, a stop transfer sequence, further into the polypeptide chain. Figure 1516. This second sequence is released from the translocation channel and drifts into the plane of the lipid bilayer, where it forms an alpha helical membrane spanning segment that anchors the protein in the membrane. Simultaneously, the N-terminal signal sequence is also released from the channel into the lipid bilayer and is cleaved off. As a result, the translocated protein ends up as a transmembrane protein inserted in the membrane with a defined orientation, the N-terminus on the luminal side of the lipid bilayer and the C-terminus on the cytosolic side. See Figure 1516. As discussed in Chapter 11, once inserted into the membrane, a transmembrane protein does not change its orientation, which is retained throughout any subsequent vesicle budding and fusion events. In some transmembrane proteins, an internal rather than an N-terminal signal sequence is used to start the protein transfer. This internal signal sequence, called a start transfer sequence, is never removed from the polypeptide. This arrangement occurs in some transmembrane proteins in which the polypeptide chain passes back and forth across the lipid bilayer. In these cases, hydrophobic signal sequences are thought to work in pairs. An internal start transfer sequence serves to initiate translocation, which continues until a stop transfer sequence is reached. The two hydrophobic sequences are then released into the bilayer, where they remain as membrane-spanning alpha helices. Figure 1517. In complex multipass proteins in which many hydrophobic alpha helices span the bilayer, additional pairs of stop and start sequences come into play. One sequence reinitiates translocation further down the polypeptide chain, and the other stops translocation and causes polypeptide release, and so on for subsequent starts and stops. Thus, multipass membrane proteins are stitched into the lipid bilayer as they are being synthesized by a mechanism resembling the workings of a sewing machine. Having considered how proteins enter the ER lumen or become embedded in the ER membrane, we now discuss how they are carried onward by vesicular transport. Vesicular transport. Entry into the ER is usually only the first step on a pathway to another destination. That destination, initially at least, is the Golgi Apparatus. Transport from the ER to the Golgi Apparatus and from the Golgi Apparatus to other compartments of the endomembrane system is carried out by the continual budding and fusion of transport vesicles. The transport pathways mediated by these vesicles extend outward from the ER to the plasma membrane, and inward from the plasma membrane to lysosomes, and thus provide routes of communication between the interior of the cell and its surroundings. As proteins and lipids are transported outward along these pathways, Many of them undergo various types of chemical modification, such as the addition of carbohydrate side chains to both proteins and lipids, and the formation of disulfide bonds in polypeptides that stabilize protein structure. In this section, we discuss how vesicles shuttle proteins and membranes between intracellular compartments, allowing cells to eat and secrete. We also consider how these transport vesicles are directed to their proper destination, be it the ER, Golgi apparatus, plasma membrane, or some other membrane-enclosed compartment.